And here we go. So session 4, species-specific ecology and pressures. Another one imagining its title, which is really hard to say for a French person. So we're continuing with you, Camille, after Axel. So it was private. Axel talked about seal populations. Camille, Duval, and after the break, we will continue with Victoria and Aikaterini, but these two other presentations will also be not online. So here you are the only online presentation of the session this afternoon. That's why also you have a lot of people in front of you. Enjoy the next 15 minutes.
Hello everyone, and thank you for being here. Welcome to my presentation on the preliminary assessment of Cozumel Islands as a potential nursery for Aetobatus narinari. So first of all, I'm going to go for a quick, I hope, introduction. So the Conductis is one of the class of the finfish opposed to the Osteichtis, and is characterized by a cartilaginous skeleton and a tigerous jaw, for example. It is subdivided into two groups, the elasmobranchs, which are the shark, rays, skates, and sawfish, and the Orocephaly, which are the chimeras. They are essential apex mesopredators, as you can see here. They live in upper trophic levels, and they control the food web structures. During an assessment under IUCN in 2021 for the Conductis, they discovered that the batteries, which are the group of the rays, are the most diverse, but also the most endangered species, with 56% of the species endangered being batteries. One of them is the Aetobatus narinari, also called the white-spotted eagle ray, that will go by step in this presentation, and it's from the Myliobatidae family. It's one of the endangered A2BD species now. And why is it important to know that? It's because this species has a vital role in the ecosystem. For example, it's an apex mesopredators, as we just said, but also because they oxygenate the water by foraging in the bottom. So Aetobatus narinari is a vital mesopredator. They live in the benthos and pelagos waters in the Atlantic Ocean, between the tropical and the temperate waters. They live in different habitats, for example, coral reef, estuarine bays, mangrove, and seagrass. And they also have highly migratory behavior, and it's a high salinity species. Moreover, they grow in nursery areas until they reach their maturity between 4 to 6 years, and their maximum size of 130 centimeters of wingspan. They also have venomous spines on the long tail to protect them from predators. So the Mexican Caribbean is one of the most critical hotspots for Aetobatus narinari and other elasmobranchs. For example, it's the second country in Conductis diversity, accounting for 26 species of rays and 39 species of sharks, including surf predators, which are the great hammerhead shark and the silver-tip shark. Moreover, the elasmobranchs take important parts of the economy. For example, it's a draw for the tourism, which attracts thousands of divers every year, but also for the fisheries, where around 4,000 tons of elasmobranchs were captured in 2017. Therefore, it's really important to understand spatial ecology, for example, for the conservation strategies application. Moreover, one of the NGOs in Mexico that is working on spatial ecology is Cozumel Research, founded in 2015 in Cozumel. They discovered in 2024 that cells have high site fidelity and high site residency in Cozumel, bringing the theory that Cozumel could be a potential nursery for Aetobatus narinari, but also for other elasmobranchs. But what is the importance of the nursery? They give optimal parameters for the growth of juveniles. Also, it's a nearshore and shallow waters, and they give shelters for the juvenile. However, with the habitat loss and degradation driven from the fisheries and the tourism, these shelters are decreasing. Therefore, it's really important to understand the concept and the definitions of the nurseries. Here we're working on two definitions that are the most likely to be applied for elasmobranch. And the first one is a Bell's criterion, created in 2007. The definition is that the nursery is a region where young are born or reside as they go towards maturity. This definition is followed by 3 criteria that we're going to talk just after. The second definition is Darwin's criterion in 2006, and it gives another type of definition of the effective juvenile habitats, which go by EJH in this presentation. And the definition is that it's an habitat where the juvenile contribution is greater than in the overall contribution of all habitats. So now the question is Cozumel a potential nursery for Aetobatus narinari? And here we're going to follow 3 objectives. We're going to set up methods. We're going to do a preliminary assessment, but also highlight the essentiality of Cozumel research and its citizen science program. Sorry. So now we're going to go for the methods and the material. Here we are in Cozumel Islands in Quintana Roo in Mexico, and it's itself an NPA, one of the 36 NPA of the Great Reserve of the Caribbean. Cozumel has itself also 2 NPAs the Arrecife de Cozumel National Park, and the Cozumel Island Flora and Fauna Protection Area. Also, in the north of the island, there is fishing prohibited area. So how are we going to apply the nursery definitions? First of all, with the elasmobranch nursery monitoring from March to July in 3 different sites in the north of the island, in Lagoon Sega principally, but also in Monte Cristo Lagoon, and then in Punta Palancar in the southwest of the island. We're going to subdivide the area into 4 stations, where we're going to put baits remoted underwater video stations, which are the GoPro associated to baits that where we will put some fish. And so this nursery monitoring is made by snorkeling, diving, using boat or kayak, but also by flying the area with drones. The second survey is the Citizen Science Program database, but also using monitoring dice. So the Citizen Science Program includes lectures and training program for the citizens, so they can be able to recognize, for example, the species, the size, and the sex, and report it on the database. Also, so this is based on monitoring dice, and the purpose of the Citizen Science Program is to follow the abundance, the distribution, but also do photo identification. So here we have 39 sites that are principally in the west side of the island. So now we're going to talk about the results and discuss them. First of all, the elasmobranch nursery monitoring. Here we had 14 surveys, which is really low, but we saw that we saw only 3 adults of 10 and 4 adults of overweight. For example, the southern stingray and the yellow stingray. Moreover, we saw that 6 of them were sighted in seagrass, and 1 of them was sighted in the flats. All of them were sighted in Lagoon Sega, which is in the north of the island. However, we did not cite anything from the drone. So here you can see that we have really low data that show weak monitoring efforts, and so therefore we cannot use these results for the definition application. Still, we validated the method with the brew and the drones because it's a reliable, non-invasive, and cost-efficient method, and also because it's a lot described in the literature. However, as you can see, there was a weak monitoring effort, principally due to the lack of resources, for example, the money, the time, the material, and the human resources. So what we can do to improve the brew and drone method, we should improve the monitoring efforts, for sure, but also take into account the buzz and kayak noise and vibration effects, and for example, the surface reflex and vibration from the drone method. So now we're going to talk about the different definitions from the monitoring types. And here we're going to talk first about a test criterion. So here, the first criterion was juveniles are more commonly encountered in the area than in others. So here we're looking at juvenile specific areas, with the first question being, is there an effect of the site on the frequency of juveniles and adults? So here in this graph, you can see the x-axis of different sites. In blue is the juvenile distribution, and in orange is the adult distribution. And as it seems here, the different sites are mostly occupied by adults. So we're going to test this with a critical wide mosquito test, with the FGO bearing no isolation between the lab stage and the site. As you can see here, the p-value is greater than alpha, therefore we cannot reach H0, and we have to conclude that there's no evidence of a juvenile specific area. However, the p-value is really close from alpha. Therefore, we think that if the data were more important, with more monitoring efforts, we could have seen this trend of a juvenile specific. The second criterion is that juveniles have the tendency to remain or return for extended periods. Here, we are looking at the global residency patterns, with the question being, is there an effect of the sites on the frequency of juveniles? Here, again, on this graph, you can see on the x-axis the different sites. And we're going to do a critical value because here, it seems that the juveniles are occupying the different sites regularly. So here, the LGO is random observation between the sites. And here, the p-value is greater than alpha. We cannot reject HCO. And we have to conclude that there's no evidence of the global residency pattern. Now we're going to look at the fourth criterion. The area or habitat is repeated use by juveniles across the years. And here, we're going to look at the global fidelity pattern. So here, the question is, effect on the site, is there any effect on the site on the frequency of juveniles at this time throughout the years? In this graph, you can see on the x-axis the different dates between 2020 to 2024, and in color the different sites. And here you can see that it seems that the distribution of juveniles is the same throughout the years, but also throughout the site. So we're going to do a critical quality test with the LGO being there is a random observation between the site over the years. And the result is that the p-value is greater than alpha. Therefore, we cannot accept H0. And we conclude that there is no evidence of a global fidelity patterns. So if you want to sum up and discuss about criteria definition, here you can see that we didn't statistically verify the juvenile specific areas, nor the global residency patterns or the global fidelity patterns. Therefore, following this definition, Cozumel is not a nursery for Aetobatus narinari. However, we saw that juvenile phylopathic tendency will not be excluded because in 2024, Isaac Weber in core showed that there is this high fidelity and high residency in Cozumel. And they use a mark recapture method, which we didn't. So now we're going to look at the second definition, which is Dalton's criteria for an effective juvenile habitat. And here the definition is that an EJH is a juvenile, have a juvenile contribution greater than the average contribution of all habitats. So here we're going to look at the different distribution between the habitats. And these habitats are coral reef, coral rubble and sands, sandflats, wall slope, and seagrass. And here you can see that the two most important habitats are the coral reef with more than 35% of contribution and the seagrass with more than 25% of contribution. And here the question is that we're going to look at which habitats have a contribution greater than the mean of their contribution, which is 20 here. And here you can see that coral reef and seagrass have a contribution greater than 20. Therefore, with this definition, we can say that coral reef and seagrass are two effective juvenile habitats. Moreover, we did a map to see the different sites that are associated to this juvenile habitat. And here you can see that 18 sites are associated to coral reefs or seagrass. 11 of them are associated to only coral reef. 3 of them are associated to only seagrass, and 4 of them are associated to both seagrass and coral reef. So here, for example, you have Palaco Garden, San Clemente Shallows, Villa Blanca Shallows, and Yucca. So if we want to discuss about Balkan criteria, you can see that here we prove that Cozumel is one of the nursery in Cozumel with the different effective juvenile habitat, such as the coral reef and the seagrass, which is aligned with the overall the white-spotted eagle ray habitats. And here, 50% of the sites are associated to MGH, which is really important to notice. Moreover, 4 of them are associated to both coral reef and seagrass, which give them twice more chance to have juveniles inside. So if you want to discuss the database citing of core and the citizen science program, we see here that the preliminary nursery assessment was valid. We show that there is coral reef and seagrass habitat that are juvenile habitats. And also we prove the accuracy of the citizen science program. Moreover, we still have weak monitoring efforts with the lack of resources, and because most of the results were given by the database reports, and that we didn't use capture mark recapture methods. So to improve this method, what we can do is to still improve the monitoring effort of core, but also improve the diving techniques and data collection by deeper training of the citizen, and also implement a capture mark recapture method with biotelemetry, such as a tag, but also photo identification. So now let's go for a conclusion. My conclusion is that the Cozumel Island is a nursery for Aetobatus narinari, so that's what we wanted to prove. And here we also prove that the method that we have are valid. For example, the roof, the drones, and the monitoring dive are efficient methods. Moreover, we saw that there is 2 effective juvenile habitats, which are the coral reef and the seagrass habitats. We show also the essentiality of Cozumel Ocean research work and its citizen science program, as the citizen science program gives us more than 90% of the results. And moreover, what we can do to improve this work is, for example, improve the monitoring efforts, but also implement capture mark recapture methods with tagging expedition and photo identification. So I hope you like this presentation. Thank you for being here. If you have any questions, let me know.
Okay, as we started with Yasmin last time, we can start with Robin in a second after the question from Mila.
Are eagle rays actually beneficial for the surrounding ecosystem?
Yes. So it's a vital. Parce que je vois pas sur le truc, je sais pas ça. So it's a vital mesopredator. So they hunt. Ça marche pas?
Si, mais...
J'ai-
Oui, mais... You're answering to a 14 years old.
Yes. So it's an important predator because it hunts on, for example, mollusk. So it's part of the food chain, and also it helps oxygenating the water because they will forage in the bottoms. So it will give more. Sorry to have the words. So it will just like oxygenate the water. So it's really important to have this eagle race. And moreover, they are highly migratory species, so they will have an impact on a lot of locations in the Atlantic Ocean, for example, for this species.
The answer is yes. Okay, Robin.
Thank you very much for the presentation. It's very nice. It's never easy to start sampling or collecting data in an area where you have hardly anything to start with. I have one question that you've started to answer, but not completely. I mean, as you went through the presentation. So when I saw your Crucial Wallis, that you have, I don't know how many sites, 40 sites or 39, something like that. In your, like, yeah, on this figure, for example, I don't know how many sites you have in total, but quite a lot. With many sites with no presence of juveniles or hardly nothing. So it's not so surprising that you don't get any strong effect because you have such a large number of sites. But since you showed that you have some, how you call it, EHG, EGH, I don't know.
Effective juvenile habitats.
That are important or that are more related to the sharks. Have you tried to run the statistical analysis based on the habitat by grouping the different sites that may be similar in terms of functionality?
So yes, I did. So yeah, okay. So here, when I did all the habitats, first I asked myself the question: is there any effect of the habitat on the frequencies of juveniles? And here is the same. I don't have any evidence of habitat preference. And then I did the same here, trying to see if the sites that are associated to an EGH are more occupied or not. And here you can see that I put coral reef and seagrass at its or EGH. And it seems like, for example, the coral reef is more occupied, not the seagrass, sorry, is more occupied by the juvenile. As here we have a peak of 30 individual. But at the end, I did a crystal quality test, and it showed me that there is no evidence of site associated to an EGH. So at the end, there's no statistics that prove that it's EGH itself.
Okay, thank you. About you mentioned the global fidelity patterns, but you don't identify the individuals. You identify the species, right?
Yes.
So when
So
you
we
see
did.
the site fidelity in general, it's for the same individual that comes back to the same area. And
So
I don't
that's why.
think that's exactly what you looked at.
Yeah. So that's why here we're talking about global patterns. Most of the time you're doing mark capture methods. That's what did Isaac Weber in core before. And as we didn't use the mark capture method, we cannot talk about individual fidelity patterns. That's why here we're talking about global fidelity patterns.
You can do that with the patterns. I don't know about elasmobranchs so much, but for some species in particular, as soon as they have distinctive patterns in the dots or in the, I don't know, say the motto, the cover, can't you identify them, or all the different individuals of the same species have the exact same patterns of coloration and dots and the... So
as you can see here, they have white spots on the back, and each spot is like fingerprints. It's the difference between each individual. That's why if we used, for example, capture method or even photo identification, it will be easier to do that. However, with the report that we have, for example, with the citizen science program, they give like photos that they put in the reports, so we can use these photos in a database. And for most of the reports, we don't have any photos. So that's really hard to use this data for photo identification. And as it's a preliminary assessment, the idea was to more see a trend and also use the method and validate the method more than really answer the question. So here I'm still happy because I showed that there is effective juvenile habitat, but it was most likely to not be able to answer the question anyway. So here, that's why I was talking about we should implement macro-capture method, but also photo identification to be able to really talk about individual fidelity and residency patterns.
Which is much cheaper to implement than, yeah. I mean, not cheaper, can be cheaper. Mm-hmm. Thank you. And I think I had the last question, but I don't, I lost it for now, so.
We can come back to you for the last question after Yasmin's questions.
Yes.
Yasmin.
Thank you for the presentation. I don't have much question. I was just wondering, how do you define a juvenile and how you differentiate them until what size? And
so, as I said, the maximum size is 130 centimeters of wingspan. Sorry. And here, for example, I did dummies of juvenile and adult. So I can see, for example, the distortion effects of the drone with the water. And so here you can see that the size of a juvenile is 30 centimeters of wingspan. And when they're born like this with 30 centimeters, it's like the minimum size. And the maximum size is like 130 centimeters.
Of a juvenile.
Of an adult. The maximum size of an adult, like when they pass 150 centimeters, they pass the sub-adult stage. So now they're getting like adult stage.
Okay, and what's your view in terms of the precision of this definition? Could it introduce a bias in your measurement?
Yeah, actually yes, because it's subjective. Like myself, I cannot always tell if it's like a big adult or just an adult of the maximum normal size. Because with the distance with the individual, they are moving also, and you have to have good practice in diving. So you don't want to harm the coral, for example. So you always have something to think about before getting the size in your head, and that's really difficult. I don't have like 100 meters for me. It's hard to say where it is. So for the size of the needle wave, certainly there is bias, and most of the time here we use mostly reports from the citizen science database. And so here it's also hard because we have to believe kind of the citizen about the size that is so yeah, it's kind of difficult.
Okay. And how do you introduce this uncertainty in your analysis? Is there a way to put more weight on value that are where you have more confidence and other where you have less confidence?
Statistically, I'm not sure. What I did is that I tried to see the pictures associated to the reports to see if the citizen that reported was kind of having a good interpretation of the size. But as I said, most of the time you don't have the photos anyway, so you can't really compare it. But what I did to put more weight into this result is that for each report associated to a photo, I will always come back to the different answer that they give. For example, even the substrates, even like the visibility or things like this, I will always try to compare the photo or the video with the reports that they made. So that's the only way I can be sure, I mean, a bit more sure about what they are saying, but you have to trust it because it's in the center. So we can do to reduce this bias is just to improve the lectures and training of the citizens. And the idea is like the NGO is kind of young. I mean, they're less than 10 years. And as there are not like a lot of people working in this NGO, they are still in lack of training with the citizen, etc. And most of the time, the citizens are coming, for example, a month every year, but they're not like training all the time. So maybe the idea is like to implement something that is really regular with the citizen, so you are sure that you're decreasing this bias, of course.
Okay. And how about the bias linked to the distribution of the measurement geographically? Did you have as many measures? Because I saw your map, I tried to guess the size. I've seen there was a legend of the number of kilometers. How wide is it? The surface area and how many measurements per, because, so we have all that sites. This is what you have surveyed, if I understood well. And if I look, it's 10 kilometers. So it's, I don't know, maybe 50 kilometers long. How long? more than that. I don't know.
What do you mean?
The whole site, what's the size? It's
I have it somewhere because I can't know it by heart. Maybe I didn't put. I don't remember. I think it's like 500 kilometers long, but maybe I'm not.
I mean, you have a scale, so we can get guesstimate.
Like you mean on the from the south to the north?
Yeah, north south, it's about maybe 60.
Well,
Yeah, 50. So I was not so far away. Yeah. So I was wondering which point you have surveyed and how many data points you had per area, because you were looking within this area, which one could be for juvenile.
So what I tried, that I didn't talk here because I'm running out of time in the presentation, but I tried to compare the number of reports with the sites, different sites. For example, here, and what we saw is that the more you go into the sites, the more you're going to have sighting, which is kind of normal.
Mm-hmm.
But that is like here, I just tried to prove that it's not just like, because when I was in Mexico, it wasn't like the real season for the eagle ray. So maybe when you see an eagle ray, it's just like a chance, no? So here I tried to prove that it wasn't just a chance. It was the more you go in there, the more you're going to see them anyway. And here you can see that the different sites are not like how I can say, like visited by the citizens at the same time. I mean, not at the same time, but in the same amount, because some of the sites are more famous, or so that includes a bias, because the citizen, when they want to go diving, they will go for the best of course dive site. And so in the historical way, that would be good for us to know it, but maybe they miss like hundreds of them at another site because it was less interesting to visit.
There are ways statistically to correct the monitoring effort, the same way as there are ways also to diminish the bias and maybe to put more weight on those data where you are sure compared to those where you are less sure. Maybe that's something you could consider introducing.
But for example, citizen science program is still something related to recreational time. So we cannot that was one of my projects, and maybe Ocean is going to work on that, I hope. But the idea was to implement something that is more regular. Like, for example, for a month, we have a lot of citizens coming in Cozumel to work with us, and so that we can have regular monitoring dive in each site every day. But that's complicated also. So, but it will be the idea.
Okay, back to you if you want, Robin, if you remember your question.
Yes, it came back as soon as you gave the floor to Yasmin, of course. Now I wanted to know how you handled the difference between observation and abundance accounts, in particular with the Brood data. Did you do present absence? How did you handle the fact that you may be observing the same individual over and over?
So that's why we put the different broods in different stations. So we divided the area into 4 stations that have to be more than 500 meters from each other. So you have kind of the time to, they are not like chasing the group, the individuals, I hope. So the idea is like if you see one individual in, like, for example, the station 1, and then 5 minutes after you see one in the station 2, the idea is that they didn't have the time to come to the station 2. So that was one of the ways we can be sure of that, kind of. And the thing is that, as I said, for example, the eagle ray, they have marks on the back that are really different from each other. So we can assume that if we see one on the footage, maybe we can try to compare them with each other to see if it was the same individual. And I think even like for the yellow stingrays or of a rays or even of a juvenile of sharks, because the idea was also to monitor the elasmobranch in general, you can always like try to find different marks that prove that it's not the same individual. But after, it depends like on the distance from the groove. It depends on a lot of things. Like, for example, also the visibility in the water. So that's bias that I didn't have the chance to work on since I don't have a lot of results on that. But yeah.
But did you? So were the I don't know, like the eagle rays attracted by the brood, or you were just seeing them passing in front of it?
So they're not really attracted by the brood. That's one of the bias of the method. As we are using Jurel and Liseta fish, they are not eating fish. They are eating. They are dirophageous animals, so they eat mollusks with a hard skeleton. And so that's complicated with the broods as they are not attracted. But maybe they're attracted by the movements. there are also that's one of the bias also in when you're doing this kind of assessment is that some individual will be attracted by what you're doing, and some of them will be like trying to go far away from you. So it's complicated, and still we don't have a lot of literatures on the eagle rays, and so it's kind of complicated also to implement it to the method.
So like
Oui,
something that came up, you mentioned putting baits when for the video. What's your view on that, and how can this?
What do you mean, the bias you can implement?
Yes, I mean, put with baits, I mean, you are attracting, and I'm wondering, yes, how scientifically this
Yes, I mean,
can be justified and
in terms of ethics, maybe? Okay. Well,
ecologically and
so in many literatures, they show that using groove is not invasive at all. And also it's cost efficient and really reliable. But the idea is there, as we just talked with Robin, the thing is that they're not attracted to the fish. So that's kind of like waste of time. But during this monitoring survey, we were also attracting the sharks. So it wasn't just about the eagle rays for this time. So that's why we still use the bait. In terms of epic, if we can talk about it, it's really important, is that we are putting, like giving food to wild animals. So that's kind of a bias also, because they will, maybe they will be very attracted to this and never get to this area, but just come here because we are here with the baits. So that's one of the bias too. The second thing is that we are working on juveniles. So the idea is that they will go for food only if they need it. So we don't really believe that they will come in this area just because we are here with the food. They will come because they're already here as we work on juvenile. Maybe for adults it will be different, but that's kind of the thing that are not really assessed yet in the literatures for elasmobranch. So it's really hard, and the idea was to test the different methods and see.
Thank you.
Okay, well, a lot of questions have been asked already. I really love that. My job is easy. Maybe can you come back on slide 16? Here you're talking about different sites. There is a lot of different sites. Is it possible? So I checked after that on the map. So all the sites are really connected to each other. They're close to each other. So have you found any pattern? Do you have any way to group the sites together according to what you observed?
So we can group the sites as I did with the different habitats.
Mm-hmm.
So as I did for the petit juvenile habitats.
Geographically, maybe.
You mean like, for example, the topography, for example? You can do like for the depth. Most of the time they are, you have a part that is really shallow because it's closer to the islands. Then if you go further, it's deeper. You can also do with the temperature. In Portsmouth, the temperature is most of the time the same in the water for different periods. So it's not really an important thing to see. You can also, so with the different buttons of the habitats, you have more or less presence of food for the eagle rays, for example. So that will be interesting also to see the food availability. And
but yeah, geographically speaking, all along the west coast, so of course, there is no pattern that you can see according to anything on the topography or on the shoreline. I know village or something where you can see like different stations performing the same.
You mean a group of sites that are having the same?
Yeah.
Maybe you can see that, yes, because some of the. I'm just trying to look for my maps. So here, so maybe you can group some of the sites because, for example, the center of Cozumel is here. So here you will have a lot of people snorkeling, diving. You will have a lot of boats because Cozumel have a lot of cruise ship. So, for example, here, the patterns of, for example, the anthropogenic activity will have a lot of impacts. And that's how we can group this.
So it's on the same regions, and you have the seagrass. So
seagrass, for example.
Okay, because here we see that geographically speaking, you have kind of groups according to the substrates. So it can also be due. I mean, what we observe with the substrate, that, for example, you have something on seagrass, can be also due to the fact that you have human impact more there than somewhere else. So.
Well, that I don't really know because I mean the sequence was here before we came with the cruise ship.
Yeah.
But maybe what we can say is that, for example, here we have a lot of coral reef, and maybe some of the coral reef are dying. I mean, they are dying. I can tell you. But maybe for some sites they are less endangered than others. For example, with the habitat degradation of the cruise ship, for example. So for some of the sites, they will have more impact of their anthropogenic activities. For the seagrass, for example, there is some areas in the sites that are like in lack of seagrass because they were like taken out from the cruise ship, for example.
On the slide 23, so when you're talking about the type of button, oh, it's fine. So maybe I will get something. So on the left, you've got the percentage of juveniles
Per habitat.
per habitats. Okay. And what is the one to the right? Is it the same, but with numbers?
So yeah, so to have the contribution, I use this short file to have the different percentage. And here, if you put the different contribution, you do the mean of this contribution, which is 20.
Why did you do the mean of the contribution? It's something else.
Here, I did the mean of this and have this.
But
And
to the left, you've got the percentage of the bottom. But 35 is what? 35 is the percentage of coral reef.
35 of the animals seen, the juvenile seen in this data are in coral reef. Whereas
Okay, but what's the percentage of coral reef compared to the?
So that we can see in these maps. Here you can see there is way more coral reef sites than seagrass, for example. That's what we can see here.
Okay. But, yeah, okay. I think we're good now. Yes. Allez, let's go for a break. So congratulations. Coming back right 15 minutes with oops. Victoria. First Victoria, yes, and Aikaterini. Allez, come back here at 3 o'clock sharp. We're going to listen to Victoria on site because online, no.
Shouldn't be so.
You again.
You know,
No one should ever.
for me,